Hello everyone, welcome back to Biotech Hub. Today we are going to start the topic metabolism. So, as we all know, that metabolism is basically the sum total of all the biochemical reactions that are occurring within an organism to sustain its life. These reactions are basically responsible for converting the nutrients into energies, into building and repairing of the tissues, and for regulating various physiological processes. So basically, we can categorize the metabolism into two broader categories, that is catabolism and anabolism. So, if we talk about catabolism, then catabolic pathways basically involve with breakdown of complex molecules into simpler ones, which tend to release energy in that process. So, this energy is typically stored in the form of ATP, which is the primary energy currency of our cell. And if we talk about catabolic reactions that are occurring in our body, we can look at glycolysis, we can look at citric acid cycle or Krebs cycle, we can look at beta oxidation. All these processes, all these metabolic pathways are catabolic in nature. Another major category of metabolism, if we'll talk about, then we can say that another category is of anabolism. So anabolic pathways involve the synthesis of complex molecules from the simpler ones. That is, it will require some energy as an input. So if we look at the examples of some anabolic reactions, then they would be either protein synthesis, lipogenesis, glucogenesis. Some or the other way, they are requiring energy for the formation. Metabolism is highly dynamic, and it is interconnected network of biochemical reactions which governs energy production, nutrient utilization, cellular functions, all those things going around in the living organism. And metabolic pathways are also essential for maintaining health, growth, survival across all the living organisms, all the biological systems that we know of. So let's start with carbohydrate metabolism. So here the carbohydrate metabolism basically refers to the biochemical processes by which the carbohydrates are broken down, converted or synthesized within an organism to produce energy and to provide some building blocks or other molecules. So carbohydrates are basically the primary source of energy for most of the organisms that we know of, and their metabolism is tightly regulated to maintain the energy, the homeostasis, and to meet the metabolic demands of the cell. So further, we'll start off with glycolysis. As we all know that glycolysis is the initial stage of carbohydrate metabolism, which is occurring in the cytoplasm of the cell. So it involves the breakdown of the glucose into pyruvate. We know that one molecule of 6 carbon glucose gets converted into 2 molecules of 3 carbon pyruvate in the process of glycolysis. So glycolysis consists of a series of enzymatic reactions that result in the net production of ATP and NADPH. And ATP generated during this glycolysis serve as a source of cellular energy. But the NADPH cannot be involved directly as a source of energy, so it plays a role in oxidative phosphorylation to produce some additional ATP molecules. First of all, starting, we know that the precursor for glycolysis is glucose. So glucose, in the very first step, the glucose gets converted into glucose 6 phosphate. So now if a glucose molecule has to convert into glucose 6 phosphate molecule, there is attachment of a phosphate molecule occurring, or phosphorylation of glucose is occurring. So this phosphate is being provided with utilization of one ATP molecule. So when a ATP molecule is being utilized, then the glucose is converting into glucose 6 phosphate. Action is catalyzed by hexokinase enzyme, mainly in the muscle cells, whereas in liver cells we have a different enzyme named glucokinase for the same reaction to occur. Now further we see that the glucose 6 molecule is being converted into fructose 6 molecule. We know that fructose is also a 6 carbon compound. So now fructose is being is a isomer of glucose, so there is a isomerization reaction occurring over here, and the glucose 6 phosphate molecule is converting into fructose 6 phosphate molecule by the presence of the enzyme isomerase, and here there is no attachment of the phosphate, so no ATP energy is required or utilized. Further, so in this isomerization reaction we are saying that the carbonyl group of the glucose is rearranging to form a keto group, which is resulting in fructose formation or conversion of glucose to fructose. Now the next step we see that there is attachment of one more phosphate group to the fructose 6 molecule, and it is converting into fructose 1,6 bisphosphate. So this reaction is being catalyzed by phosphofructokinase 1 enzyme, and here one molecule of ATP is being utilized to attach a phosphate. So furthermore, when we move forward, so this fructose 1,6 bisphosphate is being cleaved into 2 3 carbon molecules. Up till here we have 6 carbon we have 6 carbon molecules. Now we get 2 3 carbon molecules. One is dihydroxyacetophosphate DHAP, and the other one is glyceraldehyde 3 phosphate. So this particular reaction is being catalyzed by the enzyme aldolase, and now these both 3 carbon molecules, that is dihydroxyacetophosphate and glyceraldehyde 3 phosphate, they are interchangeable within themselves, help of the triose phosphate enzyme. And so the glyceraldehyde 3 phosphate subsequently gets oxidized to form 1,3 bisphosphoglycerate while reducing a NAD plus to NADH, and the energy released from this oxidation is used to phosphorylate the ADP to form ATP. The 1,3 bisphosphoglycerates get converted into 3 phosphoglycerate by an enzyme phosphoglycerate kinase, and in this particular reaction there is a substrate level phosphorylation occurring, that is direct formation of ATP, and here 2 molecules of ATP are formed because 2,1,3 bisphosphoglycerate molecules are converting into 2,3 phosphoglycerate molecules. Further, 3 phosphoglycerate gets converted into 2 phosphoglycerate by an enzyme called mutase. Further, we see that basically there is only a shifting of the phosphate group from third number to a second number position in this reaction. Then the 2 phosphoglycerate gets converted into phosphoenol pyruvate by an enzyme called enolase. Further, we see that the phosphoenol pyruvate get converted into pyruvate, which is a 3 carbon compound, which is a 3 carbon molecule, and in this particular reaction also we are experiencing substrate level phosphorylation where 2 molecules of ATP are being produced, and 1 molecule of the glucose we are getting 2 molecules of pyruvate, and there is a net gain of 2 molecules of ATP and 2 molecules of NADPH, and the pyruvate can then enter furthermore into another metabolic pathway depending on the cellular condition. What are the conditions? That is whether it's an anaerobic condition or it's an aerobic condition. So glycolysis is basically playing a very crucial, very central role in the cellular metabolism to provide energy, and it's a precursor molecule for various metabolic processes. Thank you all for your patient ears, and like, share and subscribe if you like the content.